Hello and welcome back to our YouTube channel. Today I'll be talking about eukaryotic cells and this is part 2. Okay, so as we looked at in the previous video, this is the animal cell. We talked about the nucleus, the mitochondria and the free ribosomes. We also looked at the plant cell, and in this video we'll be talking about organelles such as the rough ER, the Golgi Apparatus, the Chloroplast, cell wall, etc. Okay, so here we have some images of some microscopic images of the organelles. I would advise you to understand these and to look at these because in an exam situation it is likely that you will be asked to label these images and be able to recognize certain organelles from them. So let's start with the rough endoplasmic reticulum, the RER. It is covered with ribosomes as it undertakes protein synthesis, which the proteins which are synthesized are used for functions outside of the cell. They are transported by vesicles to the Golgi apparatus, an organelle which we'll discuss later on, for modification before they are secreted outside of the cell. The RER is often located near the nucleus, and it has a large surface area for the synthesis of all proteins. So to the right we have some diagrams of the RER and a microscopic image of the RER. Okay, so the Golgi apparatus. It is made of stacks of parallel flattened membrane pockets called cisternae. Cisternae is formed by vesicles from the endoplasmic reticulum. The proteins produced by the RER are brought by vesicles to the Golgi apparatus, as discussed before, in order to be modified. The proteins are then repackaged in vesicles for secretion outside of the cell. So the function of the Golgi apparatus is the modification of proteins which are brought by vesicles from the rough endoplasmic reticulum in order to be secreted outside of the cell. And we have some diagrams of how the Golgi apparatus looks. Vesicles. So a vesicle is a single membrane with a fluid inside. It is very small in size and it is used to transport materials inside of a cell. So as we saw before, the vesicles are involved in the transportation of proteins from the RER to the Golgi apparatus and then from the Golgi apparatus outside of the cell. Lysosomes. So lysosomes are involved in the breaking down of ingested food, organelles and damaged cells. It is important that you understand that lysosomes are found only in animals. They are dark spherical bodies. That is how they will look in a micrograph. They are a single membrane compartment, so they only have one membrane. They are formed from the Golgi apparatus and contain enzymes. They isolate these potentially harmful enzymes from the rest of the cell. And as I said before, single membrane compartment. Lyses basically means breaking down, hence the name lysosome. So yes, the function is the breaking down of ingested food, organelles and damaged cells. Vacuoles. Vacuoles are single membranes with fluids inside. In plant cells, vacuoles are large and permanent, and they often occupy the majority of the cell's volume, and they keep the cell turgid. In animals, vacuoles are smaller and temporary, and they are used for various reasons, for example, to absorb food and digest it. So they are very much like vesicles. Microtubules. So these are small cylindrical fibers which have many functions. For example, they play a role in moving chromosomes during cell division, and so they take part in mitosis, and we will be looking at mitosis in a later video. Centrioles are two groups of 9 triple microtubules. They are only found in animal cells, and they form an anchor point for microtubules during cell division and for microtubules inside flagella and cilia. The flagellum. It is only found in animal cells, and it is a thin projection, which is usually singular, which comes from the cell membrane. It contains microtubules, and its function is to move the cell around. The cilia. So these are thin projections from the cell surface. It contains microtubules, just like the flagellum, and it's used to move the cell or move fluids next to the cell. And once again, it's only found in animals. Chloroplast. So this is an organelle which is found in plant cells only, as you already know from GCSE. It is the site of photosynthesis where glucose is produced. It's made up of a double membrane, and we'll look at the reasons for why it's made of a double membrane later on in another video. It contains stacks of thylakoids, which are basically discs composed of a flattened membrane, which traps light energy. So it traps the energy needed for photosynthesis. And it also has starch grains, which may be present if chloroplasts have been photosynthesizing quickly. Cell wall. So the cell wall is an extracellular component, and it is not an organelle. And as you already know, it is found only in plant cells. It is made of cellulose, which is strong and gives support to the cell and prevents the plasma membrane from bursting when under pressure. And that's it. Please make sure to subscribe, and I hope this video was helpful.